reminder of the thermodynamics, how we calculate particular equilibria for different reactions. I think we looked at things like this. I meant to bring this slide as well, where we can plot pH versus, or maybe we saw this even before last lecture. Excuse me. Thank you. This would be called a Pourboy or Pourbaix diagram. And this is showing essentially equilibrium lines where we have 50% of one or the other along one of the other species there along each of these lines. So we could plot it as PE versus PE disinfection. This is a common thing to do. So it's what we're going to talk about today, kind of a big picture of the use of oxidation for disinfection. So we'll talk a little bit about disinfection itself, a few tangents on there, but ultimately like taking all this in the context of what's our oxidant, how are we using it, how do we make sure it's effective and applied correctly. So a bit of terminology we can start with, and actually, you know what? I'm gonna run to the restroom real quick to tackle that sneeze, so I'll be right back. Sorry about that. All right. So when we talk about oxidation, we apply some oxidant, some chemical, maybe it's chlorine, maybe it's ozone, something like that. Well, it turns out that when we add an oxidant, that is there throughout the process. So this brings us a couple of definitions here. A demand, let's say a chlorine demand or some other oxidant demand, this is some substance that is going to demand or require by reaction some amount of that oxidant before, or rather quite quickly. So before the chlorine can go do the disinfection, for example. So it's going to be a demand that is consuming the oxidant with reactions based on what's there. Then once that demand is satisfied, we have some residual chlorine or some residual ozone. So we use that term residuals to refer to the amount of oxidant that remains that can then go and do the oxidation. So we have demand and residuals, and so we could describe this in terms of the total oxidant demand would be the oxidant demand exerted at some time plus the remaining oxidant demand that is in the water. So some of these demands might be a little slower than others. We could also consider for something that's a little more reactive like ozone that perhaps it's going to just slowly be reacting away, or volatilizing away, which we could also capture with something like this. But in terms of demand, something that's actively reacting with our oxidant, that would occur over some time, and so we could look at how much is exerted, how much is remaining, and so on. So then our oxidation, might be gonna sneeze again. The oxidant demand exerted would be our oxidant dose minus the residual. So if we calculate how much was consumed, and we can do that if we want to calculate that based on how much we applied, our initial chlorine or initial ozone or whatever, minus the amount that's remaining at the end of the demand. So in some cases, we have pretty much instant demand. Something like chlorine reacting with sulfide happens very, very quickly and then would be over. Whereas some other might take some time, and we could track that according to some reaction equation where we have the rate of change of our oxidant according to, for example, a first order reaction with respect to time. Okay, so we've seen things like that. Organic matter would be a good example, whether it's natural or dissolved organic matter. Some form of it would be a good example like that. Okay, so then I'd like to talk a little bit about what is disinfection. What is the difference between sterile water and disinfected water? Yeah. Yeah, yeah.
I don't know if it's very bright, but if you can know how much you need, like the dose, why do you use like extra, like extra?
Why do we use extra, or why don't we?
Why do you use extra?
Excess for what?
Like looking at the formula given to you, maybe I really don't know that number. You put some oxidants in there, kind of use some like.
So what we're talking about when we're talking about this demand that it's exerted, it is used, but it's not used for the purpose we want. It is demanded by the stuff that we don't care about. If we think about using, let's say, chlorine to disinfect some bacteria, it's not going to take many chlorine molecules away to kill the few bacteria that are in the water. So it's actually more the case that we need plenty of chlorine molecules so that on average, the bacteria die relatively quickly for our design. But that's not really going to meaningfully change the concentration of our chlorine. Even though it is taking it away, it is making some change, it's not changing the chlorine. So it's being used, but a very, very small amount of it is actually reacting. Whereas something else that we don't care about, like the dissolved organics, may be reacting more significantly and is important there. So is that cleared up? Okay. Yeah.
Do you have to take it chlorine out and just use it?
Done with it for wastewater or drinking water, or which? Drinking water. We actually have standards we want some amount of residual chlorine in our distribution system, but we have to make sure that it's only some amount, right? It's a range, I believe it in the US, I think it's 0.5 to maybe 1.5 milligram per liter Cl2 is what we want coming out of our faucets. Yeah. Other questions? Okay. So how many of you know the difference between water that is sterile versus disinfected? A few of you may have had my class talking about it before. Okay, so if you already know the difference, don't answer, because this will be a little fun. Do you think my water in here is disinfected? If I told you it's been a little while since I washed my bottle, and I've just kind of like I pour the water out and maybe rinse it out sometimes, but it's been a little while since I've washed it. Most of you think it's probably disinfected. What about, do you think it's sterile water? Okay, a lot of you know. Do you think it's supposed to be sterile coming out of the tap? If I were to have it like a water bottle from the store, and it ideally is perfectly clean, do you think that's sterile? Some yes, some no, some little questioning. Okay. Would you be surprised if I told you that, like, the other day, like 3 weeks ago when I was in Nicaragua, I was doing some experiments with counting bacteria, and I counted like way too many on a little petri dish to count for like 100 milliliters of this water. And then now I'm drinking it. And those are supposed to be indicators of like bad bacteria, but it's probably whatever's in my mouth is what has colonized my water. So there's an important distinction here. Disinfected would mean nothing that could cause an infection. Whatever's in the water is not able to cause an infection. So we've reduced the number of any pathogens, human pathogens, to the point where it is no longer infectious. Whereas sterile refers to, so let me just write this. So no longer infectious. Whereas sterile would refer to a scenario where anything that is in it is no longer able to replicate. So you might actually have living bacteria there. You could even have pathogens in there. As long as they're not able to replicate, it's technically sterile under one definition of that term. And we use that if sometimes we will hybridize animals or crops and things, and their offspring, that hybridized offspring, are often sterile because of the genetic composition of the result, is not able to reproduce. You could use this to describe a variety of things, right? So my dog was neutered, and so he is now sterile. He can't reproduce, right? That's a kind of a technical term. Usually when we sterilize water, we are going beyond just the sterilizing, and we flat out kill or destroy all the organisms. So a lot of times it'll be like a pressure cooker type of situation. So the thing about disinfected means that some pathogens might be okay to have to consume a couple of those pathogens and it not be infectious. So if I could take Vibrio cholera, for example, and put exactly 10 of those bacteria in here and consume them, I actually have pretty high confidence I'd have no troubles whatsoever from that nasty pathogen. And that's because I have a lot of stomach acid. And just so happens that I didn't take it lunch yet, so I'm going to do a very late lunch today. So I have a lot of stomach acid. And so Vibrio cholera happens to be very vulnerable to that. Now, if it was some other pathogen, like a Cryptosporidium spore, no way in the world I'd drink that, because I'm probably now getting cryptosporidiosis, and that would be no fun at all. So this difference between sterile and disinfected, I highlight it because we want to, as we design some disinfection process, we want to understand what it is that we actually need to achieve to make water safe. It's not always, oh, sorry, I wrote here and forgot I had text already, so I'll clean that up a little bit. So we want to make sure it's not going to be infectious. We want to make sure we're designing according to what is needed. We don't necessarily have to pressure cook all of our water and make it absolutely sterile in order to drink it. So typical surface water treatment plant, we will consider many of the factors we talked about in the pieces leading up to disinfection. And then once we've got rather clean water, we've got particles out of the way, things like that, now we disinfect and we allow some contact time with a chamber. Usually this will be in a plug flow type of scenario. And then send that out for distribution, and that's where we do concern ourselves with how much residual chlorine is in our distribution system. So we will typically check the residual chlorine here to make sure that in our disinfection chamber we had the right amount, and then we might adjust it before distribution to make sure that there's the right amount. It's not too high in the first house we come to, and by the time it gets to the last house in the distribution system, it still has enough. Sometimes we'll have to have chlorine upping stations along the way in the distribution system. So the distribution system itself, all of our pipe networks and everything, are in some way part of the treatment process here. Okay, so let's talk a little bit about the different pathogens that we would concern ourselves about. Those would include, on a broad basis, viruses, bacteria, and protozoa. Some examples would be like norovirus, rotavirus, polio, and these are all food or waterborne diseases, typically. So if we're worried about it in the water, it also is quite possible that you could get it through food. So if you've ever had what you typically call food poisoning, it was probably a food infection caused by consuming something like norovirus. So if you ate something and 8 to 16, 24 hours later, you got sick, a lot of times our brains associate the most recent meal. It's not always the case. So if you have some way of identifying it, if it was like 8 to 12 hours, there's a good chance it was some virus like this. Multiplies pretty quick. Then you have a very terrible 24 to 48 hours following that, and then you're kind of better, or on your way better rather quickly. So these guys, they don't actually take too many virus particles to get you sick. It's something like 5, 10, maybe in the tens of viruses. Possibly a little lower. It depends on which one. And if you ingest them, you get sick, it's no fun. Usually these are self-limiting, meaning they will essentially, the infection itself will cause you to kind of discharge the viruses that are being produced, and your immune system will clear them if you are otherwise pretty healthy. So then we've got bacteria. And by the way, another aspect of these, we want to know what are the infectious levels, and we also want to know how sensitive they are to different disinfectants. So we're talking today about oxidation processes, but we could also consider ultraviolet light and some things like that. So viruses, they tend to be pretty vulnerable to oxidants because they don't have much protection. But if you were trying to use some filter to clean your water, they're going to escape the filter better because they're smaller. Right? They're smaller than bacteria, so they'll escape filters that bacteria would catch bacteria, but they would be more vulnerable, let's say, to chlorine. So then we've got the bacteria, things like Vibrio cholera. These guys, turns out, take something like 10 to the 6th to 10 to the 7th, so 1 to 10 million bacteria to actually give you an infection. That's because they're very susceptible to your stomach acid. And that's a great thing because, if any of you like to eat raw oysters? One brave soul. If you ever do, my recommendation is always to do so on an empty stomach, because you literally have hydrochloric acid in your stomach, so the pH is typically about 2, but if you eat a meal and it's full of food, there's still going to be some acidity, ideally, but it's going to be competing with a lot of other things and probably somewhat neutralized. And the residence time might not be the same. So if you have an empty stomach, stomach acid is nice and strong where it should be, and you have a better shot at killing all those from exposure to acid. This is also why ceviche is, when done properly, is rather protective of raw seafood or shellfish and things, because it's a highly acidic environment with the vinegar or the lemon lime juice. So Vibrio cholera, it tends to, this is a bacteria, we're going to talk more about it in a moment, can get into the water based on kind of how it plays out. So it's naturally occurring and often in the environment. Things like shellfish do, especially oysters, are filter feeding and can collect things like that and things like our viruses that might stick to the oyster flesh, and then they're concentrating a nice little tasty bite of pathogens, right? So that's the risk. E. coli and Salmonella, some of these other guys are more like 10 to the 4th or so, maybe 10 to the 5th. And these ones, sometimes you'll also have what is truly food poisoning. Food poisoning is when something in the food was poisonous. It is a chemical that poisons you. And therefore you eat it and you have a near instant reaction. If you were to take a bite of poison, it could make you sick on the spot, right? So if you were eating something and within minutes you get, quite sick, chances are that some bacteria has colonized that and produced toxins. So E. coli and Salmonella, a couple of these species are known for not just being pathogenic on their own right, but also producing toxins. So some of them are bad because they're simply pathogenic and will disturb your gut if in the wrong spot. In addition, some of them will also then be producing toxins, which makes you even further sick by poisoning you. So it's not always poison. Sometimes it's just an infection. And sometimes the infection is paired with poison. And sometimes you could have food that you accidentally let sit out for too long in a warm environment. You had some bacteria produce some toxins, and then you reheated it, killed all the bacteria, but the poison is still there. That's also plausible, okay? So things like the reason we take good care of canning foods properly is really the botulinum toxin we know as botulism. That is a potent neurotoxin. We use it for some random things like releasing wrinkles in very carefully applied doses in skin. You can like blast the nerves away so that they're no longer pulling your skin to make wrinkles or whatever. So it's a really scary poison in the wrong context. So that's another one kind of related there, the toxic side. Then we have protozoa. These would include amoebic cysts, so we don't want amoeba. We also often call these parasites in general, especially the Cryptosporidium parvum, Giardia, things like that. If you've heard of like hiker's diarrhea or something, if you drink from a stream, fill your water bottle up from a stream and it's relatively clean, but these are naturally occurring and in the environment fairly often. If you get even just, I think, something like 3 to 5 of these spores or cells will get you sick. You'll not be happy for a while. And these sometimes takes a few months to clear. So we want to get rid of those. These guys can be a little more problematic because protozoa have a thicker cell wall. They actually have a cell wall, not just a cell membrane like the bacteria. And they can also protect themselves from harsh environments by encysting themselves with an extra thick wall until the environment improves. And so they can actually resist chlorine and other oxidants pretty well. And maybe not all protozoa, but in general, and especially the Crypto and Giardia. I think ozone is fairly effective. UV is a good option, but chlorine they are quite resistant to, is the deal. And overall, the picture here for both killing them and for them attacking us is the dose makes the poison. Okay, that's a common phrase. Forgot who popularized it, but if you ingest something and it's not enough to make you sick, was it really poisonous? Was it really infective? And so with each of these, we have to consider, well, to what extent do we need to treat them? How do we design our system so that we are getting rid of enough viruses to make it not really a threat, right? How much treatment do we need in order to get the cryptosporidium down to the point where there's really one cell in a cubic meter of water at most, something like that. So we end up designing our processes thinking about what's in nature, what happens when there's a rain event and there's a lot of bacteria kicked up in the environment. How many do we need to kill in order to reliably have disinfected water? Okay, so that's kind of the thought process behind all these. And on the side note, the funny part is, how do we figure out how many it takes to get you sick? Anybody ever think of that? How would you design an experiment to see how many viruses does it take to give someone norovirus? Yeah, there's some ethical questions, right? So it turns out the CDC was, I don't know if they still do this, but for some time to study these, the ethics might be questionable. Let's say, and this is probably 20 years ago, I probably learned about this 15 years ago. So let's say you're a poor grad student, all right? Maybe even an undergrad student. And the CDC is like, hey, and this is 15 years ago type of dollars. Would you like $1,000? You'll get a free hotel, free food the whole week, and all you have to do is just take this little pill, sugar pill, might have a little something extra in it, and we'll feed you carefully and clean food, good food for the whole week. And it's going to be like a hotel type of room, and you'll have entertainment and Wi-Fi and yeah, we'll take good care of you. How about it? Oh, by the way, there might be a few noroviruses in that pill you have to take. But even if you get it, you might be asymptomatic and not get sick. And there's plenty of placebos too. You might get the placebo. You might just be lucky winner of $1,000. Anyone tempted? Anyone want to take that? Let's say it's 2,000 or 1,500, something like that. Okay, so now we're getting somewhere. Some faces changing. Like, okay, maybe. Yeah. So that's how they did it. Kind of crazy, right?
So what happens if the guest one dies?
I think the intent, their aim was to do some health screenings, make sure they're healthy, and it didn't just have to be grad students. This was just volunteers that are age bracket appropriate, that they would be healthy and should be able, with medical care on site, to not die from this, right? To not have any permanent harm was I think the way they would have gotten their IRB approval. But I don't know. I didn't hear of any horror stories of anybody dying, but it was really entertaining to think about and hear about that human testing. We might be able to get some animal testing that might be related for something that's pathogenic to both humans and some pigs or something, but in a lot of cases it's specific to the species, right? So we kind of have to, if we want to know, well, we have to try. And you kind of need volunteers because you can't forcibly, we want to be ethical about it too, right? So anyway, that's kind of the funny part to think about here. All right, so I want to talk about, speaking of understanding disease, the guy we call the father of epidemiology, John Snow. So not this guy down here. That's Jon Snow from the Game of Thrones. He knows nothing about cholera anyway. And not my brother, who has a slightly different name. John as in Jonathan. But Jon Snow. And by the way, he didn't have any children, so at best I'm like his brother or cousin or whatever's descendants. I don't know. Who knows? Okay, but he was called the father of epidemiology for his work, really being the first person to associate and identify a waterborne disease outbreak. So he associated the water, a source of water, with a cholera outbreak. So I'm going to talk to you a little bit about that. Anybody ever visit London? If you did you visit the Broad Street Pump? because you were in the e-veg class and all that. So there's a famous spot. The Broad Street pump was the source of this outbreak, and I'll describe that in a moment. So it's still there today. You can kind of visit. I've heard it's a fun. I haven't gone myself, so I need to one day. But I've heard it's kind of a neat. Is there like a museum there, or just kind of a little plaque or something?
In a street, so there's like a bunch of shops and stuff around. I think that there is like some sort of place you can go and like really see like all the stuff about like the cholera outbreak, but I don't think we went into that. We just like went to the pump and we were like, this is the pump, and then we moved on.
Okay. Okay. All right. Well, we'll elaborate on why that's an interesting experience. So this was kind of one of the first confirmations of the germ theory of disease, and the story goes that he was trying to help figure out why this cholera outbreak was occurring, and we'll explain a little more why that was such a scary, devastating moment. But he was this doctor kind of a guy, scientist kind of a guy. He had a microscope, and he took a look at the water and thought he saw some really small microorganisms swimming around, and in his mind attributed that to the illness because he was looking at it in the different waters supplies. It turns out he probably didn't have a microscope that was powerful enough to really see Vibrio cholera, so he might have been seeing something larger, some amoeba or something else, but it turns out he was not wrong in the direction, even if the actual observation might not have been accurate. It did put him onto that plus this what we call the ghost map, put him on to figuring out what was going on and identifying kind of the first person to really highlight the association between our water quality and disease. Okay, so cholera, and by the way, there's a great book called The Ghost Map. Here's the book cover. This was required reading for me. I took a couple courses in a public health department during grad school, and it's a great read. It's if any of this is interesting to you, definitely encourage you to take a look. It's like a historical account, but it was fictionalized to the point where there's like conversation that flows. It's not just like, and then he did this, and we have records that say he also visited here. It's not that. It's like fictionalized history, just so that it flows as a story. And they did really a good job with it. So it's very readable, quick read, and it's kind of a crazy, interesting story here. So turns out cholera is a pretty nasty bug because it infects our intestines, and essentially, anybody know what our like small and large intestines, like functionally what they do? Kind of remember a little bit. Obviously, digestion is happening, bacteria are growing, and sort of like a plug flow reactor, right? And so our stomachs are going to be extracting things like food, nutrients, stuff. But what else are they controlling? Anybody know? They're controlling the amount of water, okay, that is in the food that we're digesting. If we take a drink of water, it doesn't, that water will be managed, and it will be managed specifically in our intestines. So our intestines are designed largely to pull water out of the food and into our bloodstream and to achieve some balance. Of course, we have our kidneys doing it on the other end to decide how much to excrete. So our intestines, a major function is to pull water into our bloodstream. Well, cholera, turns out that this bacteria in some way attacks or colonizes the lining of our intestines, and then I don't know if it's an enzyme or a toxin or what, but their activity reverses that channel. And so now suddenly our intestines that are designed to pull water out from our food, from our gut, into our bloodstream is now pulling water from our bloodstream into our gut. So you can imagine there is severe watery diarrhea involved with any illness. The big danger is that dehydrates you very quickly. And so without knowledge of how to keep hydrated, people would literally die of dehydration within hours in some cases. And so if you can imagine living in one of the homes along this street, these black bars here, you'll see, and I'll have another image of more. Each little layer, each little blip is one death. So you can imagine how terrifying it would be to live in the squalor of London in 1850s, because it's probably a stinky, not great place to be in some ways. And suddenly, everybody in your household was fine and healthy in the morning, and by the time you're going to bed, 8 out of the 10 people living in your house are dead. Right? The horror would be, unimaginable. And that's the type of thing they were facing. And this is why a plague was so devastating and so terrifying. And so the ghost map was Jon Snow's mapping of who died where. And he put it on a map to try to identify what was going on. There's a lot of people that thought that it was miasma, which is just stench and maybe like smoke pooling in different spots from whatever industries nearby. It's just this icky sort of feeling. That kind of was their best guess for some time. So he mapped this and was able to show, well, the gross corridors might sometimes have some alignment, but it was crossing boundaries between rich and poor neighborhoods or sides of the neighborhood or whatever. And ultimately what he found was it was located around some central point. And so what he did was he added to that map where different drinking water wells were. So you see here the map tilted on its side and animated a little bit to have kind of more vertical, a little bit three-dimensional. And you can see all the deaths, right? A little more stark here. Well, the other thing you can see is right here is the Broad Street pump well. And you can see all the other flags are other wells that people might have been using. And you can see kind of how the deaths spread around. And so with this, once he had kind of assessed this and realized the Broad Street pump is probably part of the problem or the problem, he went and removed the handle from the pump. And in doing so, effectively stopped the outbreak. And there was a few more cases over the next day or two or so, but it basically petered out and died, the outbreak. And the story goes that it turns out that at the house right next to this pump, there was a family living, and of course sanitation was awful back then, so they would typically have a latrine type of thing in their home, which would typically just drain to the streets or into a pit or whatever they could manage, right? It wasn't proper indoor plumbing. And their sewage was contaminating the well water from that, which probably is not that atypical, but what was a little less typical was they had an infant who was infected with cholera. And it's important that that was an infant because infants don't have a fully developed gut system. And so what would be probably more quickly fatal to an adult, the cholera infection, was a slow going, not so fatal, and I think the infant might have even survived the whole ordeal. But a prolonged infection that was supplying this well with cholera for a long time. And so if you recall how much cholera is required to get somebody infected, you kind of need a lot of it in the environment to have this many infections. So that ultimately was the trace of the source. That baby had, I think that family had come back from some travel or had moved there from somewhere else, and so essentially the baby brought it from some other location and infected that well, essentially. So crazy, crazy story. Highlighting our need for disinfection is really one of the first times modern medicine and science kind of kind of solve that. Okay. Any questions? Did you hear this when you went to London, or was this kind of just like, oh, this was important?
Oh, we heard all about it.
All about it? Okay. What'd I
We
miss?
had this like, oh no, you didn't talk. We had this like tour guide lady who like was like bringing us around everywhere. We went to the pump and then we walked to like the locations of like the different places, which like are the same spots, but they're like, you know, buildings now and stuff. And it
Yeah.
was like, there was a lot of stuff and it was super sad.
Yeah.
You know? And they were like talking about how like, sorry to turn this into my lecture, but
No, go. No, please.
they were talking about how like in the map it was like really condensed like in this one spot, but then there was like along like the outer edges there were other like deaths, and so then they're like, well, how are people dying all the way out here? And then like also it's so like dense right here, and there was because people were like giving water from the pump because they're
Yeah.
like, oh, so good. And they're like giving it out, and so then there were like deaths around, which was also like
Yeah.
a really big like what's happening here, because they had other like water pumps in like the other locations, but like why were the other people dying? And it was like because they were giving it away.
Yeah. Yeah. Or, you know, that's exactly, I remember that was part of the book too. So whoever died out here, or like the ones over here, right? It's like maybe they were passing by and had a drink of the water, right? It's like, who knows how they got it, why they did. And in some cases you might be, you know, the hygiene was so poor. If you're caring for somebody who's sick, who got it, and you're like you live over somewhere different, but they live close, they got infected from the well, and you're over there helping and might get it from just contamination stuff, right? Yeah, but it's complicated, right? And that's why epidemiology is really the mapping of disease. That's what it's about. So just as a side note, and thank you, by the way, that's excellent. A side note, when we look at epidemiological studies, we can potentially learn things and we can potentially identify where things are going, but we need to pair that with something that we can test, because epidemiology is inherently observational. There's only so much we can do to get it close to scientific observation, and then we need to kind of pair it. Okay, so with all that in mind, let's kill some cholera, right? So chemical disinfectants, just going to go through a few of these, some of the basics here. Free chlorine is what we describe as Cl2, or when we react Cl2, let's say, so Cl2 gas, when we react it with water, we will get Cl minus, and the important stuff, HCl, of course. HClO, or HOCl is how we normally write it, if my brain will turn on. HOCl, and H+. So we can apply it as chlorine gas. That is done sometimes. Chlorine gas is extremely hazardous and difficult to manage, so it's generally preferable not to do it that way and use either sodium hypochlorite or calcium hypochlorite. You'll find this in your standard bleach at home for cleaning. And when we, of course, add something like NaOCl, we would just simply get sodium and OCl minus. Then we would recognize that HOCl will be in equilibrium with H plus and OCl minus. So anytime we're chlorinating anything, we need to navigate the fact that free chlorine technically refers to any of those three, chlorine gas, HOCl, or OCl minus. If we get really acidic conditions, we might drive the reaction back towards chlorine gas. That's pretty uncommon, but that's one reason why we do want to be careful not to ever mix household chemicals too much, because we could potentially get a situation where we're poisoning ourselves with some chlorine gas. Okay, so that's free chlorine. We'll come back and talk a little more about that, do a little bit of a refresher on identifying how much would be in the HOCl form versus the OCl minus form. This becomes really important, actually, because HOCl is a much stronger oxidant than OCl minus and a much stronger disinfectant. It turns out that I think the oxidation potential is something like 10 times different, but then the fact that HOCl is not charged makes it more capable of entering cells, slipping through where the charged OCl minus is not so able. And so it is not only a better oxidant, it is also a much better disinfectant, something on the order of like 10 to 100 times better disinfectant. I think it depends a little on which microbe we're talking about. So we want HOCl when we want effective disinfection, is the deal. The fact that this is a pH-dependent equilibrium means the pH matters a lot. So we have to understand that whenever we're designing any chlorination system. The other thing that will happen is we get what's called combined chlorine, which is what we get when we have any kind of amine or ammonia. So an amine group on an organic molecule or ammonia, we will get chloramines. Things, and I've got a slide on that in a minute, but monochloramine, where one chlorine replaces one of the hydrogens on the amine, dichloramine and trichloramine. Okay, so we have combined chlorine, and we'll talk more about that in a minute. We have chlorine dioxide, ClO2. This acts a lot like free chlorine. It's similar in potency. We do not have a protonated form to worry about. But just in terms of its impact and its oxidation potential, I don't think it's used super often, but it is sometimes used, and it will act a lot like free chlorine. We have ozone that can be used. This is naturally occurring. We don't like it down here in the troposphere because it's an irritant for our lungs. We do like it up in the stratosphere where it's going to absorb a lot of UV light. Because it can absorb UV light, we can use it as an advanced oxidant. We'll break apart and we'll have radical species that are extraordinarily reactive. Of course, as a disinfectant, we could also use UV light. That's not necessarily oxidizing, just kind of have it here with our list. And then we have advanced oxidation processes, which we'll talk more about. You already had a preview lecture from Caleb coming by. So I might adapt that lecture a little bit, might do a little more on UV perhaps instead, but we will come back and talk a little more about that. We don't normally consider our advanced oxidation as a disinfection step, but it would potentially go in the same kind of spot within our treatment train, maybe just before it or possibly just after. And that would typically be something like UV with peroxide, UV with ozone, or some other types of ways of generating really highly reactive species that'll destroy more than just bacteria. They'll get to our pharmaceuticals and things like that. Okay. The most important design criteria for designing a disinfection system would be the concept of the contact time. And I say contact time, it's not just time. It's the concentration of whatever dose that we're delivering, whatever chlorine or ozone or even UV would apply. That dose multiplied by the time that dose is exposed. So it's contact, meaning the contact of the disinfectant with the microbe, multiplied by the time of that contact. So then together that'll be a dose. And that's coming from the Chick-Watson kinetics. These are a couple researchers. I think it's Harriet Chick and I forget who Watson, but something like 1908. So really famous, long time ago researchers did great work in this field. Identified that, hey, if we do disinfection, it should follow this first order rate constant, assuming we hold the chemical disinfectant approximately constant. You can actually go look up their paper. I think it's 1908. And it's like pretty useful, readable. It's like pretty cool to see that. So if you ever wanted to dive into like really old literature in our field, I recommend it. So the rate then, our reaction would be negative K times C to the N times n to the 1. And we could describe that's the rate at which, so dn changes with dt, where n is the number concentration of whatever pathogen we're interested in. That would be in units like the number of pathogens per liter. Sometimes we'll quantify that with, let's say, colony forming units per milliliter. Okay, we'll have some metric like that. C then would be our concentration of the disinfectant. This could be a molar unit, could be a milligram per liter unit, but it's defining our disinfectant concentration. And we're going to assume that's approximately constant. If it's changing, our math gets a little more difficult, but we'll assume it's constant, and we'll come back and talk about that in a moment. So that would be a constant within our equation. This little n would be a dilution coefficient. That's the n right here. The dilution coefficient allows us to navigate the scenario where we have a non-ideal reactor, where the disinfectant is not completely mixed into the system, and there's some dilution of that disinfectant in some areas, so it's not properly dispersed. Usually, for an ideal reactor, we would just say that's 1, okay? But we could essentially discount the amount of disinfectant we actually have if it's not effectively mixed throughout our system. That's the point. Then we have a rate constant K. This would be the coefficient of specific lethality. And this is specific because it is specific to the organism and disinfectant pairing. So this would be different between chlorine and noroviruses versus chlorine and E. coli versus chlorine and rotaviruses. Each of those would have their own coefficient of specific lethality. So if we then consider, okay, let's kill Cryptosporidium, we have to consider many different coefficients of specific lethality based on the different types of organisms we know are troublesome. So if we think back on our slide where we're exploring the different doses that are required to get people sick and also commenting on how vulnerable they were, we really need to get rid of Cryptosporidium. Fortunately, they don't occur in like huge concentrations in the environment, but we still need like a 3 log, maybe a 4 log reduction of them. So we then, if those are the most resistant, it makes sense to design our system to make sure we remove those at the minimum. Many other organisms will be more vulnerable and will be killed faster. So we can kind of design this based on the minimum required disinfection for the most resistant organism. So that's the type of math we would put into it and design accordingly. Sometimes we might mix for situations like that, UV and then chlorine, so that we, some viruses are so small and kind of UV resistant that it's just less odds that they get disinfected at the dose we need or at the amount we need, but a little bit of UV goes a long way for crypto. So maybe we mix and match, and with the two technologies combined, we have confidence that we have the appropriate levels achieved. Okay, so that's the deal with this K here. And if we come back to it, and we look if C is constant, or if we make that assumption, then essentially everything before the N in this equation is constant, and we could treat those together as one constant. So K times C, we could treat as K star. This might be an observed rate constant. If we did some experiment and we observe the outcome, we would see, okay, K times C in per time units, that would be typical of a first order reaction, would give us, would match with the observations we made. So then we could write dN dt equals negative K star times N to the 1 power. So first order reaction, getting rid of that N. So this K then would just be an overall or observed rate constant, kind of a combination of those two. So with that, we can navigate these systems. what I'll note here is the coefficient of specific lethality. The units have to account for the the disinfectant concentration that will be applied. Okay, so when we look at the units, we need units overall that gives us effectively first order. But if this was a second order reaction, this would be more correct. It's because if C was changing, it would be a second order. But we put that to constant, and now we're in the first order territory. So that should hopefully make sense. Okay, as an example, this is some work I did during grad school. Essentially here, what we're looking at is using a reactive form of oxygen called single oxygen. Can make this using a photosensitizer. So this is light shining on these materials, different amounts of the materials, essentially making more of it, causing the virus to die faster. So in some scenarios, the viruses were dying very quickly, lots of our disinfectant concentration. And then what I could do is plot the change in concentration of our microbe as a log plot. So right here, the -1, that would be a 90% removal. So when we had 90% removal, or 99%, and so on, what was the CT? So we could calculate with some other things, we could calculate some estimated concentration of our single oxygen. And then if we take that contact concentration times the time that it took to get there, we could find, okay, let's place this value. And here's a different experiment, and here's a different experiment, and so on. And so the point was to ultimately derive some plot and a line here that showed, okay, from our observations, our CT followed approximately this. So you'll see CT plots with some frequency where they would then take information like this and plot, let's see, CT on the x-axis, and then they would have, what am I thinking? They typically put on the y. A lot of times you'll see plots, and I should have this, and maybe I have a slide on this, where you'd say, okay, here's a line of 99%, or here's a 3-log removal line, and plot it that way. It might actually just be simply the log of time versus log of concentration. Okay, anyway, I'll get you a better plot on that. I should include it if I don't have it. But anyway, that's the point, is to find the K and then essentially parameterize our disinfectant, how good it is, what's the impact for some situation. So how do we identify, how do we count? Well, here are a couple of examples of culturing bacteria, allowing them to grow in order to count how many viable bacteria there were. So in some cases, we could pour some on a plate, spread them around, and then we count X number of colonies on a plate. We could also pass it through a filter. So this is not the exact one, but when I filtered some through my water bottle, or from my water bottle, it looked something like that. Pretty gross. So you should try that sometime. Actually, I need to do an experiment and clean it. Now that I'm back and I've used it, I should test it some more. Anyway, each of these little globules, and the bigger ones, those are essentially bacteria colonies that began presumably as one bacteria, or maybe one little cluster of bacteria that landed on a spot. So to get really highly quantitative information about our disinfection process, we try to use direct colony, or as we'll see for viruses, plaque forming tests. There are some other things you can do that indicate some average number and require some, you'll get some most probable number with some statistics, but the gold standard for disinfection is to have countable plates where you say, okay, we started with this number, we diluted it a bunch, so we found 10 to the 8th bacteria per milliliter or whatever, did our operation, and now there was 10 to the 3rd. So that was 5 log change. So that's how we like to do it with disinfection. For viruses, you can do something kind of similar, especially for we use indicator viruses often, something like a bacteriophage. You grow up a bunch of bacteria, whole blanket of bacteria across this, add in your viruses, and each time you see a little dot here, there was a virus or some cluster of viruses that began infecting the bacteria that were growing, caused some bacterial genocide, and destroyed lots and lots of bacteria right around that little spot. Because one cell would burst, spread to the neighbors, they would get infected, and so on. Probably worse than cholera if you're a bacteria. Anyway, so then you can count how many viruses are there. You can do the same with tissue cells, potentially, for if you needed to do something that was not just an indicator, maybe you're doing a real pathogen or something. I don't ever go to that extent because that is requires a lot more biosafety and requires a lot more time for each of these cultures, a lot more difficult, and a lot more funding. So the nice thing about a virus that infects a bacteria is that you can get results rather quick. You don't have to wait a few weeks and keep the cultures going for weeks at a time. But just showing you can do a variety of things to test how many, in this case, plaque forming units, PFU, per volume you have. There are some other tools we can use, some that are maybe semi-quantitative. Recently I've been working with a couple of colleagues on looking at ATP analysis. If you take cellular matter that has energy molecules, ATP molecules, you can extract that and then add an enzyme that causes it to luminesce, to shine light out based on how much ATP is there. And that's an interesting quantification. You can get some quantity of ATP with a pretty simple measurement, but that's going to include everything, not just pathogens. So a lot of these indicators or counting don't really get there. Now you could also go a little more specific and find a particular organism that you are interested in, do some DNA analyses, and I think some of you in this room working with Dr. Bivens or Dr. Mo probably could give a better lecture than I could on the DNA side of things. But the point is you can get somewhat quantitative and specific to an organism. So that can be quite helpful, especially if you're looking for that organism in the environment. The disinfection side of things, we normally default to species that we know have a similar K, if we drop back to our coefficient of specific lethality, similar K to the most problematic microbes. And something that we can easily handle that's not going to infect us in the lab when we're using it, and design accordingly. But it would also be very important to know, in some cases, well, what is in our water? Is that particular microbe there? Some of that epidemiological investigative analyses would want tools more like these. Okay, so that brings us to an example, put a little of this to use. And I'm going to give you kind of two pieces of this that we could look at. We'll just go with the simple form of it. I'll give you a few minutes to work on it. So here we have data for HOCl disinfection of poliovirus at a concentration of 1.8 milligrams per liter. That would be our HOCl. It is shown below. It's adapted from some study in 1979. The temperature was 20 Celsius, the pH was 6, and the task is to determine the rate constant of inactivation assuming Chick-Watson model applies with an N value of 1. And this should be in just a batch type of situation. So you're given data, and this happens over the course of 8 seconds, exposed to 1.8 milligrams per liter of chlorine. Okay, so I'm going to give you a few minutes to work on this. I'm going to go use the restroom, and then I'll talk to you a little bit about how to handle the chlorine and what assumptions might be reasonable. So see if you can get an answer or see how close you can get to the answer And perhaps if you find that you might want to make some assumption to simplify the process, go ahead and write that down and see where your thoughts can take you from there. That would be a useful practice. So I'll be back in a minute. Well, that was, I think my cord just touched my keypad, and that just sent it to the end. Okay, try again. I'll be back. Yeah. The Chick-Watson model said dn dt is equal to negative k c to the n power times n to the 1 power, and you can derive from there to a consider it in a batch scenario, you do the mass balance from there. Mm-hmm. Okay, I'll be right back. Yes. All right, so hopefully you had some progress. I'm going to just highlight a couple things real quick and then let you spend another minute working on it, if you'd like. This provides us some information we could use or could decide not to use. We have temperature given. We were told 1.8 milligrams per liter of HOCl, but if we add HOCl directly, should be in equilibrium with OCl minus. The question's actually not super clear. So maybe we're talking about straight only HOCl, but then there's a question of why they mentioned the pH in the first place. or the temperature. If we wanted to account for exactly how much HOCl is there compared to the total, we could find that using our equilibrium equations, find the appropriate rate constant for this equilibrium reaction. It's going to be somewhere near 7.5. I think 7.54 is a good default for 25 Celsius, so it's probably somewhere pretty close to that for the pKa. And then we could solve using 1.8 milligrams per liter as total chlorine, and then find the HOCl fraction at pH 6. We could go through all that and find some real HOCl concentration. Or given these conditions, maybe it would be nice to just assume that approximately 100% of chlorine is HOCl. And if we were to do the math, it's probably like 98+%. So it's probably, if the question is really asking us to do this, it would probably be 1.78 or something milligrams per liter of HOCl that's really there. So for the moment, I think it might make sense to assume that it's 100%, and we'll come back and practice that type of thing next time anyway. So assuming that, consider what the next steps would be that you would need in order to get to your answer if you haven't already. And hopefully what you notice is you're given data, and you could plot that, a number of concentration of viruses per time, and it would probably look something like this. Because it's a first order reaction, and if we do the mass balance, which I have kind of elaborated over here, this should be n equals n naught times e to the minus kt, which would give us something like that. So then our task would be to linearize it. We learned earlier, take the natural log of n over n naught to get it in the minus k star t form. Then we can plot that in Excel or graphing calculator or something. And this slope should be our k star. K star times 1.8 milligrams per liter is our k, our coefficient of specific lethality. Rather, k star divided by c. So that would be our approach to finding. And the question wasn't even really clear. Maybe it was asking for a k star because it was saying the rate constant of inactivation. In that case, I would probably give it both because it looks like we could do both, but so does that make sense? All right. So that's it for today. We will do a little more on the disinfection side of things on Thursday and continue from there.